Um, Good evening to all the participants. I think I'm audible to you. Yes.
Yes,
Uh, yes. Good evening to everyone. So let me start the session 11 on T cell epitope prediction. Okay. So we have completed 10 sessions up to present. So before going to session 11 on T cell epitope prediction, can we recap what exactly did we discuss in the previous session, that is on concepts of reverse vaccinology and immunological databases? So what exactly did we discuss in the concepts of reverse vaccinology and immunological databases? Can you discuss the last session topics? What are the last session topics we have seen? Yes. So I got immediate answer from Veera Rahul. We have discussed something about acid tool, an online tool, which is used for reverse talking. Very good. And we also discussed something about T and B receptors. Okay, so what is the shape and uh, shape of T cell receptor, and as well as we have seen MHC molecules, isn't it? We have seen shape of T cell as well as MHC molecules. We have also seen one such example, that is dendritic cell interacting with the T cell. And we also seen MHC1 molecule. If you see the MHC, that is major histocompatibility complex, the major histocompatibility complex or the genes, like it vary with respect to person to person. The MHC or protein complexes. So related to MHC genes, if you speak, we call these MHC genes as HLA genes. MHC is protein, and genes wise we call it as human leukocyte antigen, HLA genes. Okay, so that's what we have discussed. If you see the MHC major histocompatibility complex, there are three molecules, 1, 2, and 3. Okay, 3 is something related to complement. It tells about the chaperone, sheet shock proteins, etc. But in MHC1 and MHC2, you find major genes responsible for providing a kind of immunity in terms of cellular or humoral. Okay. And if you see this, especially we have a cellular immunity with the MHC molecules. And if you see the vaccinology, we have discussed about what is vaccinology and what is reverse vaccinology. Okay. And then we have discussed adjuvants and the importance of adjuvants. Very good. So I got answers from Veera Rahul, Nandini Loganathan. We also discussed about types of actions she has commented, and Purple World Rose about vaccinology and MSc genes. Good. Jayashri about significance of adjuvants. And again, from Purple Rose, IEDB database. Okay, so IEDB databases. So participants join on time. Why are you joining 5 minutes late? Join on time. For many participants have joined 5 minutes earlier. So you won't miss anything. Okay, if only if you attend from the beginning, then you can understand what exactly we recapped and what we are continuing. This is the chapter 11 is a continuation of chapter 10. Chapter 10, 11, 12 is basically about vaccine design. So basics of vaccine design we have seen in last session. And now we come across with a particular uh, T cell epitope vaccine prediction. You know, if you see this T cell epitope based vaccine, uh, I have published a paper. I think even yesterday I was mentioning that a paper on Nipah virus T cell vaccine, T cell epitope vaccine. It's not just a T cell vaccine, T cell epitope vaccine. I told you one more information on yesterday that T cells itself can be used as a vaccine. Not only T cells, dendritic cells can also be used as a vaccine for combating cancer, for combating infectious virus. Okay, so why I'm showing infectious virus is most of the viruses are linked with cancer disease. Most of the viral diseases are linked with the cancer diseases because we have a kind of a comorbidity. If a patient is more prone to the viral infections, obviously they may uh, get a kind of a cancer based on the kind of a viral infection. So all these things has to be checked from patient to patient. Okay, if you see also one more book chapter I published related to enzyme inhibition applications in treatment of human viral diseases, and also I published one more paper on cancer treatment. Okay, enzyme inhibition applications on cancer treatment in a book. So if you see this book chapter, you get some idea. So before that is just a review paper, and now in this slide we can see the workflow of how this T cell epitope prediction was done. I told you that we have plenty of structural, non-structural proteins. We have taken protein W of Nipah virus uh, from the Nipah virus genome, and then we have predicted so many antigens. Plenty of antigens came when we predicted from the IADB tools. Then we just screened the best epitope. Only one epitope we have chosen for MHC1 for H and one more epitope we have selected for MSC2, and we try to talk with the MHC molecules, and whatever molecules we have dropped, we finally take it for simulation analysis. RMSD, RMSF, radius of gyration, other parameters are determined. And I told you if CD4 T cell. So I think I can interact now. What is the other name for CD4 T cell? What is the other name for CD4 T cell? There are two types of cells, isn't it? One is majorly cytotoxic T cell, the other is helper T cell. These cells are interacting with MHC molecules. So if I tell CD4, the CD4 T cell interacts with what molecules? Interacts with what MHCs? And what is the other name for this? What is the other name for CD4 T cell? So I asked two questions. CD4 T cell, that is CD4 marker in T cell, is going to interact with one MHC. What MHC is that? MHC2? MHC11? No. APC immune cell. Of course, it interacts with APC immune cell. But what kind of protein marker in antigen presenting cell is going to interact with CD4? MHC1, T helper cells, helper T cells, MHC2, MHC1. You got confused, is it? So I can tell you once again. Okay, so now don't confuse. This is MHC1. Okay, in my slide it is wrongly mentioned as MHC1 here. This is MHC2. If it is MHC2, it interacts with am I right? CD4 is interacting with the CD4 is interacting with MHC, so it is cytotoxic T cell. Cytotoxic T cell. This is cytotoxic T cell. This is helper T cell. So in T cell, you have helper T cell, and the other is cytotoxic T cell. So CD4 T cell is a cytotoxic T cell, and CD8 T cell is a Th cell. The helper T cell generally interacts with the MHC2, and cytotoxic T cell is interacting with the MHC1 molecule. Okay, don't confuse this. We have only we majorly discuss about these two types of T cells, but there are different subtypes of subtypes in T cells. In T cells, we have different subtypes, but generally for MHC1 and MHC2 interacts with TC and TH. That you should remember. You see the helper T cell, still we have further subclasses like TH1, TH2. Okay, so majorly in helper T cell, you have a CD8. It can be TH1 or TH2, but we have a different marker for TH1 and TH2. This is how we can distinguish even in these types of helper T cells. Got it? So like this, we have natural killer T cell. It's not shown in any of this MHC1 and MHC2. Okay, we also have a native T cell. We have regulatory T cell. We have tolerant T cell. Okay, so like this, we have so many kind of T cells. Each and every T cell, whatever I mentioned, has a unique marker. Like how I tell, a helper T cell has a CD8, and cytotoxic T cell has a CD4. Likewise, even the other T cells have a unique marker. Okay, so this is how we can distinguish, these molecules has to interact with these kind of just interacting with CD8 with the MHC to won't give a kind of a signaling. It won't activate the T cell. If you see the signaling, especially the activation of T cell, you need at least 2 or 3 interactions. The most significant is the CD8 T cell receptor. CD8 T cell receptor interacting with MSC molecule, and the other is CD8086 molecule of antigen presenting cell is interacting with a CD28. If only if it interacts with CD28 of T cell, then only the T cell gets activated. But instead of the CD80 or 86 of a dendritic cell or AP antigen presenting cell, if it interacts with CTLA4, cytotoxic lymphocyte antigen 4, then it's going to suppress the T cell. It won't activate the T cell, but it's going to suppress. So the suppression is needed in terms of autoimmune diseases. Okay, this is also we have seen on yesterday. Okay, what do you mean by co-stimulation? This is what I call it as a co-stimulation. The CD8086 or the co-stimulation markers of APCs. Okay, so the co-stimulation has to be done for T cell activation. But we also discussed about the T cell receptor. We have seen about T cell receptor. I told you that in T cell receptor you have molecules. Okay, nearly 4 molecules. 4 molecules of CD3. Okay, surrounded by the T cell, you find in the form of a complex. But in 2 dimension, you can see only 2 CD3 molecules. Okay, so likewise, we have also seen the antigen, the epitopic peptide of MHC2 molecule is of MHC2 molecule is large. How much large? 13 amino acid in size. I told you in John Hopkins University, if you see some Coursera courses, they mention that better use these kind of a size. Okay, and if you design the peptide for cytotoxic T cell, that is especially to bind with the MSC1, you can have 9 amino acid epitopic peptide. And we have also seen this kind of a DC-TH cell interaction. And if you see the dendritic cell markers, other than these markers interacting with T cell, we also have nearly 162 markers on the surface. We also seen one such mechanism, that is MHC1 mechanism. How an antigen presenting cell is going to present an antigen on the MHC surface. Okay, there is a mechanism for it. So what is going to happen in antigen presenting cells, especially dendritic cells, directly go and combat the cancer cells. If it sees the cancer cells, it directly go for combating. If it combats, obviously the proteins or whatever released, okay, will be taken by the antigen presenting cell by phagocytosis. In the APC, you have a proteasome. So the protein get inside the protein degradation enzyme called proteasome. You get the proteins are degraded into peptides. These peptides are transported through transporter associated protein. Then later it in the endoplasmic reticulum. The participant can mute. One participant has, yeah, who is that? So I think one participant has unmute. Kadir Velu sir, today also. I think he has muted. Okay, now let me take to the session. So if peptides are transported to the transported associated protein, so these peptides of antigen, cancer antigen, is going to bind on the surface of the MHC molecule. What molecule? MHC1 molecule. And it's going to transport through the Golgi complex and then further transported through the secretory vesicles, finally transferred to the surface of the APC, where you can find that red color antigenic cancer antigen protein. If this cancer antigen protein has to be detected by T cells, and even if T cells want to combat the cancer, then the T cell has to interact with the antigen thing. So in T cell, whatever you are going to have a kind of a receptor, here you have a paratopic region. So this epitope is going to bind to the paratopic region of a T cell receptor, and obviously the T cell gets a kind of a memory to combat against a particular cancer antigens. Whenever it finds, not only this APCs can go and try to kill, but also T cells also is going to have a memory to combat only the particular foreign cancer antigen. And similarly, structure of MLC molecules. In MLC molecules, we have 1 and 2, and the shape-wise, we find a difference. We find alpha 1 and alpha 2 on the surface in class 1, but in class 2, we have alpha 1 and beta 1. Just because of this, the peptide, the epitopic peptide length is somewhat larger. So both these class 1, class 2 molecules are supported through the transmembrane segment. And if you see the MSC1, 2, okay, so basically you should remember that in class 1 you have HLA-A, HLA-B, and HLA-C, okay, in the chromosome 6. In the same chromosome, class 2 MHC, you have DP, DQ, DR genes, okay, and in class 3, we have majorly complement TNF, tumor necrosis factor, and other heat shock proteins. So differences between MHCs, I told you we can able to study in the table in any immunology textbooks. You can find this information. Like however I mentioned, there is a pathway for MLC1. We also have a pathway for MLC2. Okay, so there also, if any cancer antigen is found, the genetic cell takes this antigen through the phagocytosis, and then it try to project this in the MHC2 surface. But we have a different pathway for this. Okay, and if you see this HLA complex, HLA is the gene name. Actually, MSC we generally describe for a protein. And if you see the HLA A, B, C, I told you in the chromosome you have a short arm and a long arm, isn't it? So the HLA A is present in the short arm and C and B in the large arm, and even this DP, DQ, and DR are in these locus regions. Okay, so if you see this major MHC class 1, you have HLA-A, HLA-B, HLA-C, and if you see the minor MHC class 1, you have HLA-E, FG. So not only you have ABC, ABC are the major genes of MHC class 1, but even you have minor genes like EFG. And similarly for the class 2, we have DP, DQ, and DR, but also for the minor region, you have DM and DO. Okay, but if you see the databases, majorly we discuss on these genes, HLA, A, B, C, and DP, DQ, and DR, each and every allele. For example, the person from Tamil Nadu may have more HLA, TP. The two in HLA, TP, we have subsets like HLA, DP 0101. Okay, if Tamil Nadu is HLA, DP 0101, may the West Bengal, if you see the alleles from the West Bengal, definitely we have a phenotypic variation, isn't it? So even if they have HLA-DP, but they won't have the same 0101, but they have something like 0301. Okay, we have some subsets where we can distinguish the alleles through the numbers. Okay, so all these things we can see in the website. Okay, so not only we can see based on the states in India, we can also see the alleles based on the communities. The Nadar community have one allele, and the Mudaliyar will have one allele, Chettiar will have one, Yadav will have one allele. So likewise, we have different alleles. If you want to design a kind of epitopic peptide for treatment of cancer for a specific community people, even that is possible. Okay, that is possible using reverse vaccinology. Okay, now let me take you to the HLA information later linked with different diseases. One such example I'm showing for the autoimmune. Even HLA molecules are linked with the cancer. So if you see these autoimmune diseases, HLA molecule, HLA-B27 linked with spondylitis, reactive arthritis, acute anterior, and HLA molecules B47 is linked with the 21 hydroxylase deficiency. And diabetes mellitus type 1 systemic lupus SLE is linked with HLA-DR3. So likewise, if you see, yeah, the participant can unmute. Yeah, so if you see these HLA molecules, yeah, participants, kindly mute. So if you want to question, you can question. No one is questioning me, yeah, even at the end or in between. But why? I don't know why you're unmuting. Okay, let me take you to the PPT. So if you see the HL alleles are or linked with a particular autoimmune disease. Similarly, we can also see for the types of cancers. And if you see this kind of Nipah virus genome, I was explaining that even viruses are linked with cancer diseases, viral diseases, or linked. If viral diseases are linked with cancer, I just am showing you with an example, Nipah virus genome. So you should, if you are focusing on liver cancer, you should study about hepatitis B, hepatitis C, D, E viruses. Not only B and C or the major viruses linked with liver cancer, but also the other but they have a less role. The DE is having a less role. Okay, so likewise, okay, so each and every cancer is somewhat linked with viral viruses. But this is a kind of infectious virus, okay, Nipah virus. And if you see this is something about structural, non-structural proteins of Nipah virus genome. I just targeted the non-structural protein tablet because the other proteins are already studied. And so just because it's already reported in the journal papers, it made me to choose like this. But nowadays, if you want to work on reverse vaccinology in a particular virus, I think most of the research is completed. Most of the research completed, and they are published in a repeated journal papers. So the left out is to work on the cancer. If you want to work on cancer, you can use the reverse vaccinology, and you have to check even how you can link with a particular virus. Okay, the viral. So that also you have to check whether you have any research work pending or not. Okay, let me take you to the IEDP database. So in IEDP database, you have epitopes parameters. You have assays, T cell, B cell assay. Not only you have T cell, B cell, we also have MSC ligand assays, MSC restriction assays. Like for example, if you want to discover a epitope with respect to MSC class 1 alone, you can select it. Even class 2 alone, it can be selected, or any MSC2 like the epitopes are designed based on the parameters whatever we select. And if you want to design an epitope, if you want to screen an epitope based on a particular disease, even you can type the type of disease and you can get the information on that. Okay, if you want to get the epitopes for a particular disease, you can type a disease. So not only you can enter the infectious, allergic, and autoimmune, even you can type any type of a cancer here. And if you click find, obviously the epitopes, the cancer antigen epitopes are shown. Okay. And here, if you want to use T cell epitope prediction, we have plenty of tools. Here we have one MSC1 binding, MSC2 binding, MSC1 processing. In the MSC1 processing, we have proteasome and transport associated protein. So these activities are determined through MSC1 processing. You know, if you see the MSC molecules, you have two types of functions. One is antigen presentation, the other is antigen processing. If you see the antigen presentation, as earlier mentioned, in the MSC pathway, so the antigen is presenting in this step. And if the antigen has to present on the surface, okay, it takes few more steps. And finally, the antigen is presenting on the surface. So initially the antigen is presenting here, but if only if it exposes to the surface, then only it can interact with the T cell, isn't it? So for the final presentation is happening on the surface of the APC. Okay, that is MLC on the surface of APC. And the processing is something like If only if the antigen is interacting with the antigenic epitope is interacting with the parotopic region of T cell, then only you can find a co-stimulation is going to happen. Signaling is going to happen. Then only the T cell gets activated. So this is processing. If this is presentation, and only if the T cell interacts, we get processing. Presentation and processing are the two important functions. Okay, so that's what I want to mean. So processing, if you are speaking about presentation here, the presentation is something related to binding, isn't it? The processing is something related to the activity of the proteasome and tap. So based on these parameters. And MS1, immunogenicity. If you want to check whether the epitope, whatever predicted, is immunogen or not. So there is a difference between antigen and immunogen. Okay, immunogen majorly your antigen property is to specifically bind with the antibody, isn't it? But immunogen, which gives an immunity on binding. Okay, there is also a biomolecule like an antigen, but this here the major application is to give an immunity after the binding. So the MSC1 immunogenicity can be predicted using hyperlink. Okay, so this take you to the tool, and from the data whatever deposited here already in IADB, it searches and it produces with the results. And similarly for the B cell epitope, we have antigen sequence properties can be determined using the link. So more about the B cell epitope we'll be seeing on the next session. So today we cover about the information, what are the information we can get from the IETB database, and what are the tools we can use in T cell epitope prediction. If you see the T cell epitope prediction, okay, the T cell epitope prediction methods are broadly divided into two classes. One is direct method, the other is the indirect method. If you see the direct prediction procedures are based on the T cell epitope sequence and structural analysis. Got it? Direct prediction is based on T cell epitope sequence and structural analysis. It means it uses features such as ampipathicity motifs that bind MHC's molecules. So, but these are quite inaccurate and highly false positive, whatever we predict directly. There are also indirect methods which predict the binding of the MSC peptides by using some of the smart techniques such as machine learning techniques like artificial neural networks, support vector machines, and the other supporting theories. Okay, so using the machine learning techniques, we can predict the indirect, you can predict the binding of MSC that we call it as the indirect method. Okay, and if you see the prediction of cleavage and transport associated with the antigen processing are also the crucial elements for the T cell epitope prediction and are related to T cell immunogenicity. This is one such parameter we can determine from the tool present in IEDB database. If you see this T cell immunogenicity, how this immunogenicity is calculated. So whatever we are telling one peptide, a peptide and it's a small uh, protein. It's not exactly a small protein, small polypeptide. அதாவது like it will be like 9 to 13 amino acid in a sequence. You don't have any other molecules, you have only amino acids, isn't it? So if you want to check that 9 to 13 amino acid from immunogen, that is antigen, what we call it as an immunogen, is having immunogenicity or not. For this, you have to use these three parameters calculation. Number one is antigen processing. Number two, peptide binding to MHC molecules. Number three is recognition by a cognate T cell receptor. So these three parameters are important properties for the immunogen, which is predicted using this tools.iedb.org immunogenicity. Okay, so we have a tool in that database itself. And similarly, we can also find the conservancy analysis. Okay, like the conservancy is the fraction of the protein sections that the epitope contains an identity is correspondence. Like if you have a similar kind of thing like you're working for SARS-CoV-2 train, sorry, if you want to discover a vaccine, epitopic vaccine for a SARS-CoV-2 strain, but that you want to check, you want to design for delta kind of a strain. Okay, but you already have a vaccine peptide for alpha strain, and whatever epitope vaccine you got for alpha strain, if it works for delta strain or not. You want to check, okay, even these kind of similarity information also we can check in the conservancy. So what is the major difference between the alpha SARS-CoV-2 and the delta SARS-CoV-2 strain? The major difference is one or two amino acids in the spike glycoprotein is mutated. The other proteins, I told you nearly 4 structural proteins are there in SARS-CoV-2 and 16 non-structural proteins in SARS-CoV-2. If you see the other 3 structural proteins and 16 non-structural proteins remain same in both alpha and delta. Only one or two amino acids alone are mutated in spike glycoprotein in these different strains. Sorry, in these strains. Okay, so this we can check through the conservancy analysis. Okay, so after prediction. So there are two types of calculation. One is linear analysis and non-continuous sequence conservancy analysis. So this is the URL. And if you see the toxicity, even the toxicity is determined. How a software is determining a toxicity, you know, the toxicity of a chemical compound or toxicity of the predicted peptide, all these things are calculated based on the physicochemical properties of that particular molecule. So what are the physicochemical properties majorly we take for calculating the toxicity? So we are using the hydrophobicity, hydropathicness, also called as hydrophilicity, molecular weight, net hydrogen, net hydrogen donors and acceptors both, and then side bulk and steric hindrance. Side bulk and steric hindrance. All these parameters are important to calculate whether a compound or a peptide is toxic or not. Here, we generally use whatever the website URL mentioned. This is used for predicting the toxicity of the predicted peptides. So, whatever peptide you are going to predict from the EDB tool, that should be immunogenic and that should be non-toxic. These are the desired properties we anticipate. Okay, I think I already told you how to go for. So initially what I'm going to do in objectives, if you say first I initially have to predict the peptides from the tools, okay, from the IADB tools, and then you have to dock dock the peptides. I think this is not right. I can tell you while I explain. Okay, so what are the open source softwares used? In the subsequent slides, you can get in detailed methodology how I work. So now we can see the materials and methods. The open software used is IADB. That is in IADB, we have tools NetMSC PAN and NetCTL PAN, which is used for predicting MSC class 1. And for predicting MSC class 2, we are using MSC class 2 server. Immunogenicity, I think I've shown you all these things in the IADB website itself. For B cell prediction, we can use this algorithms to predict the B cell epitope. And for toxin prediction, we can use for toxicity analysis. And for getting detailed annotation of a protein, you can get from UniProt. And appropriate for MSC class 2 prediction. An Autodock winner for docking purposes. Discovery Studio for docking analysis. So if a peptide is docked with a protein, this analysis can be done even using the Discovery Studio visualizer. Yeah, I told you first you have to predict the peptides, then you have to check for immunogenicity, then you have to check for toxicity, then you can check for conservancy. So these are some of the properties we determine. So this is one such Nipah virus W protein I have taken from UniProt. So this is the UniProt ID, P0C1C7. So I have taken this W protein, and then what I did, I have initially passed it to these tools. I passed it initially to the NetMSC PAN, NetCTL PAN, ProPred, and IADB. So IADB is only one website, but also I want to check with the other tools. These are other private servers. IADB is a different server, and these three are three different servers. These three are having different URLs. So why I'm trying with different tools? The objective of trying with different tools is to get a concurrence. If I just work with a single database and single tool, whether it can tell that is the best peptide, so we can't able to justify. So what I did, I just predicted the T cell epitopic peptide from the other tools, from the other servers also, and then just I got the concurrency, which is from which website to website is matching with each other. Okay, then I got according to this course. Okay, for Nipavirus W protein for this peptide SPVIA I got this as maximum score, that is 0.8, and we got here as 0.566. Okay, so I have chosen this as a base peptide. But whereas if you see the other peptides, actually the net MSC percentage rank should be low. Okay, and net CTL band should be this range. So this made me to choose this kind of a peptide. So to know more about this, it can get the information from the journal papers I mentioned there. So also, if you see the immunogenicity, conservancy, and to these parameters, you got a convincing results for this peptide. So not only just uh, screening from the 4 different servers, but also I checked for these 3 important parameters. Finally, I docked the peptide with the HLA molecules. So whatever score I got from the Autodoc, I just shown here. Okay, and also I've taken this for simulation analysis. Okay, let me take you to the website and then I can start explaining about this, then you can understand. Then we can come to the importance of adjuvants and how the adjuvants can be discovered. First, we can see how the epitopic peptides can be discovered. So for going to the epitope database, you have to type ietp.org. So this is the URL. Okay, and if you want to predict the peptides for a particular cancer, okay, for example, if you want to find for breast cancer, so I'm typing breast cancer. So you're getting the types breast cancer, carcinoma, etc. You select one thing, okay, whatever you want, and you just click find. Because in India, the maximum number of cancer cases are there in this type. So I just open this. So if I click that disease name, okay, here they're asking to search further. You can search like that, or you simply click this. So you got some more information on this. Mm. So related to breast cancer or current selection, apply. Search. So I just selected breast cancer and click search now. After clicking search, I got nearly 5043 antigenic epitopic peptides. So these are antigen epitopic peptides. SIIGRLLEV. If you get nearly 5043, what is based out of this 5043? If I want to select only one, how we can select? So you have to use machine learning algorithms. Okay, you have to use machine learning techniques to screen the base peptide. Of course, you have to look through what kind of antigens you are searching for. There are classes of antigens, isn't it? So whatever epitopes you have predicted, these are derived from the different antigenic proteins. So if you're focusing on a particular kind of, for example, if you're focusing on heat shock protein 90 beta, for this is one of the peptide. So for this IADP ID is allotted. So you have, if you mention this IADP ID in any of the IADP tool, it can able to understand. So how many assays are performed, how many references are provided for this. So all this information is present for that specific. So if you see the epitopes, how many epitopes are there for breast cancer? 1,26,000. And antigens are 13,675. Assays, 1,40,000 assays are there. 9 major receptors are there. Okay. And if you see this references, major references, you can find like this. Okay, so journal it take you to the journal paper. So like this, you can search for any type of diseases. Here we got infectious, allergic, autoimmune, isn't it? Other than this, we can also search for any kind of cancer diseases. So not only we can search like this, okay, but we can also search based on the class 1, class 2. For example, if not our community population has majorly HLA-A, this kind of asterisk 0201, you can type the same, okay. And if you want to, HLA-A comes under class 1, isn't it? So either you can click a class 1 or you click any and you can search the corresponding epitopes. For example, HLA-A, if I want to select this HLA-A, I'm selecting this. And so if you want to select that particular gene, you can select like this, or if you want to select a particular MLC1 class 1, you can select this. Now, if I want to type, the server is slow. Yeah, HLA. If you want to select the only one, you can select this, click find. You will be having that, but simply click search. If you click search only for that HLA-01 thing, you will be getting so many epitopic peptides. I think the server is slow. You'll be getting information. Okay, it takes more time. Okay, let me take you to the few more tools. Likewise, you can select for assays if you want to get assay information. If you want to get epitopic information for a particular organism, not only you can work on human, but you can also work on virus, plant. Okay, so because even the plants have viral infections, isn't it? They face various diseases. If you're working on plant biotechnology, you can if the genome is published, obviously that kind of a genome information are also deposited in this. So you can search for that kind of an epitopes also here. So one such example they provided is a peanut. Okay, so type the Python sativa or peanut and you simply click find and get some information on that. So you get epitopes for diseases by searching through diseases or searching through host. If you want to get only for human, select only the host, human, or even you can get not only in the mouse, but you can also get for any other animals, dog, camel, feline like cat. So all these informations for all these species also will be getting corresponding epitopic peptides. Okay. So, okay, let me take you to this application of the tool. The first tool, what I'm going to show is the T cell prediction, epitopic prediction. We have MLC1 binding, isn't it? So, I'm going to click this MLC1 binding. Okay, initially I told you for MLC1, no, not MLC1, for Nipah virus W protein, this is the sequence. That's what I mentioned you, isn't it? So this is the sequence we got from UniProt. So what you have to do, you have to enter protein in say FASTA format, okay, or as wide separated sequence. They're asking to paste in the FASTA. So the entire, even you have to take these identifiers, which comes under FASTA format. Simply paste this. So now this is in FASTA format. Okay, so after pasting the protein in MSC1 binding tool in ADB, or even you can download it as whatever I copied. You just have to paste it in Notepad and save something like file name .fasta. And however you say file name .fasta, simply choose that file, you upload that file, and you can search the epitopes for it. Okay. So defaultly here, if you see the database, it uses a prediction method like NetMSC 4.1. There are also methods like 4.1 BE, consensus, artificial neural networks, SMM, PVC. So there are multiple kind of methods. So defaultly, whatever method we use is the recommended. So I'm not going to change anything. Now, of course, we want to check, we want to discover or design vaccine peptide for human, isn't it? So I'm not going to change human. I restore human as the organism. And now But remember, this is the protein I have taken for the virus. I am checking compatibility with the human. Whatever epitope I'm going to, epipeptide I'm going to predict, I'm going to check for the human. So I kept like human. And now allele. In allele, you can find HLA01. So there are plenty of alleles. For HLA-A itself, you have this much number. And after A, you have B and you have C information. There are plenty of alleles. So to what population I want to design the epitope, for that alone you have to get, you have to select. Okay, that alone you have to select. So to know more about uh, HLA alleles, you can also get information from this population coverage. There is one option called population coverage in the IEDP tool. Click this, it take you to this. So here you can find ethnicity. So in the world, East Asia, Northeast Asia, India, but if you want to design a peptide for India, you have to select India. Okay. So like this, you can select, and there is also one database called allele frequency net database. You have to click inside. So in that population database, go to this allele frequency database. So inside that, okay, you can find populations. Click this populations. So if you click this populations, you get browse populations by geographical regions. Click this. Now it take you to the information. Okay, here you can able to see based on the geographical regions, HLA molecules, KR molecules, cytokines, MICs. So for each and every region, this much information is deposited. This much information is deposited in IEDB database. Okay, India comes under which region? South Asia or Southeast Asia? If it comes under Southeast Asia, we have 195 alleles, HLA alleles, because we are working on HLA alleles, isn't it? MLC1 and MLC2. So you have to click here. If you click there, we are getting China, Hong Kong, Malaysia, Singapore, Taiwan, Thailand. No, it's not in Southeast Asia. I think India is in South Asia, 62. Yes, India is there. So we got the Indian population in South Asia. In South Asia, if you see this Andhra Pradesh Brahmin people has majorly HLA DRB1 only. And if you see the Delhi people has these alleles TPV1, TQB1, TRP1. If you see Kerala, Nair, maximum population, ABC, they have HLA ABC. So these are the sample sizes mentioned here. But according to the sample size, what are the major genes identified? Okay, so for a specific community also, we can able to design the vaccine peptide, okay, for a type of a cancer. So likewise, okay, so for all the kind of ethnicity in India itself, we are getting some information on the major genes. So by selecting these genes, we can able to make it out. For example, if I want to work on Andhra Pradesh Brahmin, okay. Now, what I'm going to do, I have to click this India Andhra Pradesh Brahmin. So in Andhra Pradesh Brahmin, you get some information on this. Okay, we also have allele frequencies. I'm going to click this allele frequencies for the Andhra Pradesh Brahmin. Nearly 98 sample size is there. Okay, if you see this Andhra Pradesh Brahmin, I got majorly TRP1 0, 3, 4, 7, 8, 9, up to 16 you have. Okay, so the maximum percentage of individuals in the DRP107. DRP107. But what is DR? DR is MHC2. DR, TP, DQ comes under MHC2. So they didn't provide much information on MHC1, but they provided information on MHC2. And for the major MHC2 is TRP1. So instead of MHC1, if I am going for MHC2 binding, you can click this MHC2 binding and just paste the W protein FASTA format. And what I told you, whatever you're going to select, the DR is the one major thing, but in allele, what is the thing you have to select for the Andhra Pradesh Brahmin? For the Andhra Pradesh Brahmin, we have found out that majorly they have TRP107, isn't it? So TRP10107. So now this allele is loaded. And if you want to take the second major allele also, okay, so the second major information is for DRP115. Okay, so based on the percentage of individuals, DRB15. So I'm going to select this also here now, DRB10115. So these two alleles are got selected now. So now whatever vaccine peptide I'm going to get for Nipah virus is for the Andhra. You can able to cure Andhra Brahmin, but I don't know about Tamil Nadu Brahmin. I don't know about Kashmiri Brahmin. Okay, but for this is more effective. So this is what we call it as a precision vaccine design. The epitopes are predicted based on the specific alleles of geographical location, isn't it? And if you're going for the MSC2, I told you to select the 13 size. Okay, if you the 13 size is preferred. Okay, so it can select the 13. Okay, I've selected two alleles already. Okay, and now if you want to provide an email address an option, you can submit it, and then you have to simply submit. Now MLC2 binding prediction is happening now. Okay, in Brahmin we have only MLC2 as the major. We can take some other where we can find even MHC1. Now we are searching only MHC2, isn't it? If you see MHC1, you can get even for the Delhi, India, Delhi. You have ABC. Even Andhra Pradesh Kolla is having AB. So you can choose anything. If you want to select some kind of MHC1, choose this India, Delhi, POP2. Okay. So here you have allele frequencies. So here you have, which is the major one? So the 20% of individuals have this gene, A0101. So if I want to predict MHC1, okay, so you have to simply use HLH0101. The length generally preferred is 9, isn't it, for MHC1? So that's it. We already pasted the W protein, selected a default database, human, a specific dominant allele, provide an email address, click submit. We have already clicked submit for MSC2, isn't it? We got an output for this. See the result. For MSC2, we got this much results, this much peptides. How many peptides you want to know? If you want to know, you can simply download the result in Excel format. So if you click the Excel format, you can able to find the result. So bottom also you find this link to download. So many peptides are generated. Okay, thousands of peptides are generated. But these peptides, you select the best peptide based on the percentile rank. Based on the percentile rank. Okay. So here they might have mentioned which is best. High score is good binders. High score. Which is high score? This is higher score. If you want to sort it out based on the higher score, click here. Click here once again. So this is the best peptide we got. For MLC2, based on the score, this is the best peptide. Okay. And similarly, we can also check for MLC1. Now the MLC1 is going to predict epitopic peptides. We also got plenty of thousands of results. So if you drag from top to bottom, thousands of results are there. Download this Excel sheet. And if you want to see which is the best, high score good binder in net MLC pan of IADP tool and IADP database. The NetMSC is a tool present in IITB web resource, isn't it? IITB database. So according to this high score, which is high score? This is the high score. So the best peptide is this. Likewise, however, I am telling in NetMSC1, MSC PAN, and NetMSC2 PAN, there are also a few more websites. Like I was telling you something about NetMSC, NetCTL PAN, isn't it? I've also used these tools in Nipah virus epitope prediction. So you have to, in Google, type NetMSC PAN and simply get inside. So it take you to this kind of a website. Here also you get an option to paste the FASTA format, and also you can select the MER peptides. If you want to select 9-mer, select the 9-mer, select the alleles, and simply click submit. If you click the submit, obviously you get a similar kind of a results, but the results won't match because these have a unique algorithms. They use a different parameters to calculate the uh, antigenicity, that is antigen presentation of a peptide. So, but you have to check which matches and which is going to have a maximum score. That is the best one. So you have to cross check with NetMSC PAN. You can also check with NetCTL PAN. Also, there is one more server called ProPred. I told you there is also one more server called ProPred. So there are plenty of tools in immunoinformatics to predict the epitopic peptides. You can select at least 3 to 4 tools. In some papers, they might have used 7 different tools to predict the best T cell epitopic peptide. Here, I have taken only 4 tools. So, ProPred is another tool. This is used for predicting MSC class 2. So, likewise, even for MSC1, we have other tools also. Simply paste the sequence, select the alleles, select the thing, and finally click the submit. Okay. So, this is how you can able to take it for antigen peptide uh, calculations. Okay, and if you go for processing, this is another tool. Till now, whatever you have seen is for MSC1 binding and MSC2 binding, but we also have 2 more tools. So one is MSC1 processing, the other is immunogenicity. But I have also taken you to the population coverage to show you the alleles. Okay, now go for the T cell epitope prediction. I told you for the peptide binding, we have already the tools. Even the links are here. Even using these links, you can predict the peptides. Other than this, we also have TEP tool. The TEP tool provides prediction of peptides binding to both class 1 and class 2 molecules. TEP is for both class 1 and class 2. And other than this, for T cell epitopes processing prediction, we have proteosomal cleavage, TAP transport, MSC class 1 combined predictor. Neural network based prediction of proteosomal cleavage sites, NetCHOP, and NetCTL and NetCTL PAN. So this is available here. And MHC-NP, this is for prediction of peptides, naturally processed by the MHC. So likewise, we have tools even for MHC2-NP. Not only we have tools for the processing, I told you we also have tools for immunogenicity prediction, isn't it? So the immunogenicity can be predicted by clicking this hyperlink. Deimmunization, CD4 T cell immunogenicity prediction, antigen expression based epitope likelihood function prediction, T cell receptor match, LYRA, lymphocyte receptor automated modeling, structural complexes of epitope receptor, TOP, which is used to intend to allow the para MLC1 modeling. This tool has been developed by TOP. So this much tools are available. So if you want to predict any particular property of the T cell, that is a property of the T cell epitope, then use any of these tools. Okay. Of course, even if you click this immunogenicity, it take you to this. You can predict the immunogenicity. Okay. So the next session we'll be discussing about how to predict the B cell epitopes, and we also discuss about how to go for. I can also show an example like how to predict the immunogenicity. And then what are the other parameters to be taken care for epitopic prediction and vaccine adjuvant preparation, how to prepare the vaccine adjuvants. So, okay, all these things, the adjuvant design, etc., all these things will be seen in the next session on tomorrow. So, in this session, if you have any doubts, you can ask me. Do you have any doubts? So, today I have covered about the T cell epitopic prediction through MLC1 and MLC2. Okay. Tomorrow we see nearly 7 algorithms for T cell epitope prediction. Okay, participants. Glad to see that even today you have nicely interacted with me. So thank you. So see you on tomorrow.